Hello students, today we are going to start the last part of the chapter Neural Control and Coordination, that are the different sense organs. We have already completed the neurons, the introduction of the neurons, then the types of nerves, types of neurons we have seen. Next we have seen the conduction of nerve impulse, RMP, AMP, transmission of nerve impulse. Then after that we have seen the different parts of the nervous system, that is CNS and PNS. So under that we have seen the structure of the brain, function, then sympathetic parasympathetic nervous system. Then after that we have also studied about the cranial nerves and the spinal nerves. So we will start the last part of this chapter now. There are different sense organs. So including the eye, we are going to have 5 sense organs. So all of the sense organ have one main basic function, that to carry the stimulus, carry the senses, convert the different types of energy into electrical impulse. Then after that send this impulse to the brain. Brain will perceive the stimulus, then produce the response, and that's how we can able to coordinate with the environment, with the surrounding. Right, so let's just start first of all the structure of the eye from outside externally. Then after that we'll move to the internal structure. Then we will see the mechanism of vision. So first of all, this eye, roughly it is around 7 grams in weight. Okay, it is oval in shape, right? It's oval in shape. Next one, where it is located in the skull. We have the socket, and in the socket, the socket is called as the orbits. So in the orbits we are going to have the eyes, right? Now from outside, if you see the protection of the eyes, okay, so we will see first of all protection. So if it comes to the protection, see, this is the eye, okay, so this part, this will be your cornea, okay. Here we are going to have a layer, that is cornea. This white part will be called as the sclera. This part will be called as cornea. Then here this color, okay, this is because of a very important part actually. This is the iris, okay, and inside the iris we have centrally a hole that is called as the pupil. So this is just the structure. Above the iris we have a transparent layer that is called as cornea, right? So remember this thing. So first of all, what are the protections we have, right? So just see what is it. See, first of all, here, suppose here we have the nose. So here we will have a red spot, right? So what is that red spot? Actually, these are the 3 eyelids we have. Okay, now we understand only the 2 eyelids, right? So apart from upper eyelids and lower eyelids, we have another eyelid, that is the 3rd eyelid we can say, that is called as the nictitating membrane. Okay, so this is the nictitating membrane, which is vestigial. Okay, vestigial means it is not functioning in our body. So it is actually one horizontal flap like this, and it is going to work in the other groups of vertebrates like fishes, amphibia, reptiles. And yes, it is also present in the apes, but it is gradually decreasing its function. So in human it is vestigial completely. Okay, this actually helps the organism to see underwater. So this is with the help of nictitating membrane. But in the air atmosphere, this nictitating membrane do not have any function left, but as a vestigial organ it is present. Now this is the 3rd eyelid. Now we have another eyelids also. So what are they? These are the upper eyelid, and this is the lower eyelid. So here we have the upper eyelid, okay. Now only the upper eyelid is movable. If you can think about it earlier or not, I don't know, but upper eyelid is movable only, not the lower eyelid. So this is the lower eyelid which is fixed, we cannot move the lower eyelid, right? Look, only the upper eyelid is moving vertically, which helps to clear the surface of the eye and also makes it moist. Okay, so this is the lower eyelid which is immovable. Okay, remember. Next thing what we need to know, that above what we are going to have the eyebrows. Okay, so here we are going to have the eyebrows also. So what are the functions of this eyebrows and eyelashes? That it is going to protect our eyes from the direct exposure of dirt or some external other factors. Okay, dirt, pollutants, that or sweat even are not directly allowed to fall on the our surface of the eye. So to certain extent the eyelashes and the eyelids will prevent the entry of, or prevent the flow, or prevent the dust or some foreign particles to fall directly on the eyelids, on the surface of the eye. Now after that, see here we are going to have different glands also, right? So these glands are also protecting our eyes and to keep the eye surface moist. Okay, so what do they see? Towards the base of the upper eyelids, what we are going to have one gland. So this gland is called as the gland of G's. So this gland of G's, and we have another gland that is called as meibomian gland. Okay, so this meibomian gland and the gland of G's, they are actually the modified oil gland. Okay, so these are the modified oil glands, so that this, whenever we are blinking, every time this oil can keep the surface of the eye moist, okay, and prevent water loss. So this is the oil gland which is present on the base of the hair follicles of our eyelids. Next we have another gland. Here also it is present on the eyelid surface itself. What is that? This is called as the gland of mole. Okay, so this gland of mole is actually a modified sweat gland. Okay, it is the modified sweat gland which is also producing or secreting the sweat on the surface of the eye to keep the eye moist. Okay, next thing, another gland we all know, that is about the lacrimal gland. Okay, so what is the function of the lacrimal gland? We know, all of us, we know this is the tear gland. It produces the tear. Okay, it is tear producing gland. What is the composition of tear? What the tear composed of? Tear basically contain water, different type of salts, and some bactericidal secretions, enzymes, that is called as lysozyme. So if any bacterial infections, bacteria enters or fall into your eye surface, then this tear, it's not like whenever you're crying only in that period of time you produce tear. No, every time, constantly, every time we have the secretion of lacrimal gland, that is tear. Whenever we are in an emotion, we are sad or we are crying, then the secretion of tear gland increases. But every time we produce that tear, which is actually helping us to prevent any bacterial infection. So that tear contain water, then salt and lysozyme. Okay, so this lysozyme will kill any type of bacteria. So another thing, another very important layer we have. See, here what we have, the white part is the sclera. This part, which is the colored part, that is the iris. In that iris we have a hole that is called as pupil, through which the light enters inside the eyeball. And just to protect this iris, we have the uppermost layer, that is the cornea. Cornea and sclera is a singular layer. Okay, so this is going to be the outermost layer. Now here above the cornea and sclera also, okay, so this is the sclera, this is cornea. Above it also we are going to have another layer. What is that? This is called as the conjunctiva. Okay, so this is the conjunctiva we have. Okay, so there are some infections, that viral infections can cause the purplish or pinkish eye, or we can call it as the conjunctivitis. This is actually the infection of conjunctiva, so that's why it's also called as conjunctivitis. So suppose this is the sclera, this is the cornea. Okay, this part is the cornea, which is protecting the iris, and here this is the white part, this is the sclera. Okay, above that what we are going to have, these are suppose the hair follicles we have. Okay, now here this one is the conjunctiva. Okay, even it is not allowed directly any type of foreign particles to fall on the cornea or sclera. Above the cornea and sclera also we have the another very important layer, that is the conjunctiva, which is the first layer. Now, why it is important to make the surface of the eye completely moist, or why it is going to have this oil or sweat secretion, why every time lacrimal gland is producing the tear? So the main reason is to keep the transparency. Okay, so here what we have above the iris, we have the cornea. Cornea is the transparent layer, all of us we know. And whenever it says, like, we suppose some individual is donating his eye, so it's not the whole eye will be transferred, it will be just actually the cornea which will be transferred from one individual to the next individual. So there is the cornea which is replaceable, which can be replaced. Okay, so the thing is, in the cornea we don't have any blood vessels, so that we can make the cornea transparent. Okay, so if there is no blood vessels carrying nutrients and oxygen to the cornea, then how the cells of the cornea can survive? So the thing is, directly from the atmosphere, from the surrounding environment, we get this oxygen. Okay, so cornea will get the oxygen directly from the outside, from the air. So that's why whenever we sleep for suppose 10 hours, 12 hours, our eye become red. Why? Because in that period of time we are not getting sufficient oxygen. Okay, and to whenever we wear lenses for long period of time, also in that period of time we will not get proper oxygen supply to the cornea, right? So cornea, to maintain the transparency, what happens, we have the moist layer, then only the diffusion of gases can be possible. And that's why actually we're going to have this gland of G's, meibomian gland, which are the modified oil gland, and then the gland of mole, which is a sweat gland, and lacrimal gland, which is providing the sweat and oil mixture, as well as the tear gland produce tear, so that the surface of the eye can be moist, right? So after that, let's just move to the structure of the eye, right? So let's just see, this is just the outer structure. Now after that, we'll move to the structure of the eyeball in detail. Let's just move to that part. Now after that, let's just see the structure of the eye. So I'm going to draw the eye. Let's just see from first. So first of all, this eye is oval in shape. Okay, so see, this is suppose the eyeball here. So this is the eyeball. So first of all, here we have the outermost layer, right? So what is the name of this outermost layer here? Okay, so this outermost layer has two parts. Okay, posterior fifth sixth part is called as the sclera. Okay, it's the sclera, which is the posterior fifth sixth part. Okay, so this part will be called as the sclera, and here in the front what we have, this will be the cornea, which is the anterior only 1/6 part. Okay, so I have told it about this this cornea just earlier, that above the iris we have the transparent layer, that is cornea. Cornea operation is 100% successful. Always cornea operation will be successful. What's the reason? Because it is not connected with any blood vessel. So easily without the cut of the blood vessel, we can transfer the or transplant the cornea. Okay, so this is the outermost layer. The second layer, what is it? So this is the second layer. It's not a complete layer. Only the outermost layer is a complete layer, but here this will be the second layer. We have, what is the name of it? This is called as the choroid layer. Okay, so this is the second layer we have. So this second layer is called as the choroid. Okay, anteriorly it is going to have some processes, and those processes are called as the ciliary process. Okay, so here this is called as the ciliary process. Okay, so here we have the ciliary process, right? This is the ciliary process, and posteriorly ciliary processes is going to have some muscle in it. Okay, so this ciliary processes posteriorly, it is going to have a muscle that is called as the ciliary muscle. Okay, so this is the ciliary muscle we have. Okay, now after that, attaching to the ciliary processes, we have some ligament, and that ligament is called as the suspensory ligament. So here we have some ligament that is called as suspensory ligament, and this suspensory ligament is going to hold the lens. So here we are going to have the lens. Okay, so let us see here, this is the lens through which the light will enter inside the eyeball. So this is the lens, okay, which is going to focus the light on the retina. So that part will move. Now the next part, that this same ciliary process will going to have one extension. Okay, so here it is the extension, right? So this is going to have different color depending on the pigment we have. Okay, this is genetic. So what is this? This is going to be called as the iris. What I have showed, that above the iris we have a layer of cornea. So this is the cornea, and here it is the iris. Okay, so this is the iris, and in this iris, centrally we are going to have a hole. So this is the hole. This hole is called as the pupil. Okay, so this hole will be changing its diameter so that we can ensure how much light will enter inside our eye. That will be the diameter of pupil. It is going to change, right? So above this, what we are going to have, the next very important, the innermost layer, which is the sensory part of our eye. Okay, so what is this layer called? The third important layer we have. What is this? This will be called as the retina. Okay, so this is the innermost layer. We have the retina, which is very much essential. So here in the retina we have the photosensitive cells, and those are called as the rods and cones. We have rods are sensitive to white and black vision, and then the cones which are sensitive to the darker light, brighter light, not darker, brighter light. So it is sensitive, or it's going to give us the colored vision. So this is retina, we'll come to that. Now these are the three layers we have, right? So in the front region, what we are going to have, so here we are going to have the hair follicles. You know already this is suppose the hair we have. Now here, to protect even the cornea, what we are going to have, the outermost layer. What is this? Above the cornea, I just said about this. This is called as the conjunctiva. So this is the conjunctiva we have, right? Now in this eyeball, you can see there are two different chambers. Okay, so what are the two chambers we have? So just see here, this is one chamber. I will just put the dots. So here one chamber we have that is called as the aqueous chamber. So this is the aqueous chamber we have, and then here this is the another chamber we have. Okay, just draw it a different color. So what is this? This is called as the vitreous chamber. Okay, so this is the vitreous chamber. Okay, so this one is the vitreous chamber. So both these two chambers, they're filled with two types of liquid. The aqueous chamber is filled with the aqueous humor, and this vitreous chamber is filled with the vitreous humor. These are the two types of liquid. The aqueous humor is transparent, okay, and this aqueous humor is secreted by this ciliary processes here. And then this vitreous humor, that is going to be a opaque. It's not completely transparent, it is opaque in nature, and which is going to refract the light. Okay, it's going to refract the light because when the light is passing from one medium to the next medium, the angle is going to change. Okay, so the refraction of light occurs in the vitreous chamber so that we can focus in the one point of retina. I will tell you what is that. So this is in the retina, it will focus and it will be helped by this lens. So this is the lens here. So what is it? See, aqueous humor is produced every after 24 hours. Okay, so every after 24 hours we are going to secrete the amount of aqueous humor. But the vitreous humor is going to secrete only once in our lifetime. That is in the embryonic state whenever I was formed. Okay, now this aqueous humor, which is, suppose on the earlier day we have produced that much, which is going to give nourishment and the moist environment to and protect the nourishment or give nourishment nutrients to the lens, then iris, cornea, everything will be given nutrients and a moist environment by this aqueous humor. So this aqueous humor is produced every after 24 hours. So excess of aqueous humor, if it is there, then there is a canal we have. Okay, so here we have a canal. So this canal is going to take out or remove the excess amount of aqueous humor every day if it is in excess. So what is that called? This is called as the canal of Sclera. Okay, so this is called as the canal of Sclera, which is going to wash out the excessive amount of aqueous humor if it is present in the aqueous chamber. Because if it is happening that every day we are producing the aqueous humor every after 24 hours and earlier day the remaining amount of aqueous humor is not moving outside or it's not being used up, then every time the here in this aqueous chamber the pressure will start to increase. Every time the amount of aqueous humor will start to increase if we do not drain the excess amount of aqueous humor of the previous day. So that's why excess amount of aqueous humor which is remain unused, that will be drained outside by this canal of Sclera. So there is one disease also which is related to the blockage of canal of Sclera. So that is the disease we will discuss, that is called as glaucoma. In glaucoma what happens? The canal of Sclera gets blocked, and that's how every time, every after 24 hours, every day we are producing aqueous humor. So remaining amount of aqueous humor of the previous day will get accumulated, and gradually time by time, every day after day, this pressure in the aqueous chamber will increase, and that gives more pressure to the cornea also, the lens also, right? So this is one actually a condition, disorder we can say, that is glaucoma. That is the reason of blockage of canal of Sclera. That's why this disease occurs. Okay, so this is the structure of eye. You can just pause it and you can remember it. Now posteriorly what we have, this is the optic nerve we have. So this optic nerve will take the nerve impulse from the eye to the brain. So here in this region we are not going to have any type of photoreceptor cells in it. So here this spot is called as the blind spot. Okay, and here in this region of retina there is one specific part we have. Okay, so there is a specific part we have, what is that called as? This is called as the yellow spot. Okay, so in the yellow spot you can see in the middle there is a depression. So what is that depression present? That is called as fovea centralis. Okay, so there is a depression. Normally the light focus on the yellow spot. Why it is in the yellow spot? Because it is going to have maximum cornea in it. Okay, so maximum not cornea, maximum cones in it. So maximum cones are going to be present in the fovea, and as I already have told you that the cones are sensitive to uh, the bright light. If the light enters fall on the cornea, the cornea, it will pass through this aqueous chamber via this lens, via this aqueous chamber, it will fall on the yellow spot. Then after that, this fovea, in that fovea we have the cones more. So these cones are going to give us the colored vision, right? So here otherwise we have rods also, we have cones also in the retina. The cones are going to give us the colored vision. When the light is proper, there is bright light, we can able to see, or we can able to see visible the different colors or shades of light. But when the light is not enough, in dim light, timid light, we can see only the black and white vision. What's the reason? Because in that period of time only rods are working, and the rods are sensitive to the bright, that is the black and white vision. Okay, so this is the black and white vision. When the light is not enough, the rods are working. When the lights are enough, then the cones will work. So this is the structure of the eye. The eye is going to have three layers. Outermost layer is the sclera. The sclera is the posterior 5/6 part, and anterior 1/6 part is the cornea. Middle layer is the choroid, and the innermost layer is retina. Outermost layer, that is the sclera, which is going to have the protective function. It's made up of white fibrous tissues which going to give us the proper shape to the eyeball, and also the eyeball will be protected by the sclera. The choroid is the nutritive layer which have the blood vessel. It is going to be connected with the suspensory ligament, which is going to connect with the lens as well as the iris. Excessive light, if it falls inside the eye, it is not allowed to get reflected. So excessive amount of light will be absorbed by the choroid layer. Then the innermost layer, which is retina, which already we know that it is going to be sensitive to light because it is going to have the cells, rods and cones, which can able to give us the different colored or black and white vision. So retina is the photosensitive layer. Otherwise sclera and choroid also are the layers which going to give us the shape and help us to give nutrition by choroid layer. So this is the structure of the eye. Now after that we will see the mechanism of vision. So till now you just make a screenshot or just see this picture, pause it and draw the picture by yourself. You will understand easily. So let's just move to the next part, that is the mechanism of vision. How can we see? Let's just move to the other part. Okay, so after that let's just see the mechanism of vision. So first of all, whenever it comes to the mechanism of vision, then we need to know the structure of the retina. So in this mechanism of vision, we'll see the different sublayers of retina. So retina is going to be divided into three sublayers: photoreceptor cell layer, bipolar cell layer, and then the ganglionic cell layer. So let's just see, suppose if it is the outermost layer, I have already drawn the structure. You can see this is sclera. Then the middle layer, what is that called? This is the choroid layer. So this is the choroid layer. And what is the choroid layer? And what is the innermost layer called? That will be called as the retina. Okay, so here this is the innermost layer. So this innermost layer, suppose this is the retina. Okay, so this retina is going to be divided into three sublayers. So what are the sublayers? So the first sublayer here we have, this is the first layer. this is the second layer, and this is the third layer. So the first layer here, this is the first layer we have. So what is that first layer we have? That is called as the photoreceptor cell layer. Okay, the second layer, what is this called? This will be called as the bipolar cell layer. Okay, what is the third layer we have? The third layer is called as the ganglionic cell layer. So this is the ganglionic layer. Okay, so we are going to have three layers. What are they? Here, this one is the photoreceptor cell layer, middle layer is called as the bipolar cell layer, and then the innermost layer, that will be called as the ganglionic cell layer. Now, questions can come from this part also, that you just need to arrange the layers from outside to inside or from inside to outside. If it is from inside to outside, then first of all ganglionic cell layer, then bipolar cell layer, and then third layer is the photoreceptor cell layer. If it is from outside to inside, then outside we have the photoreceptor cell layer, which is attaching to the choroid layer. Then outside the choroid we have sclera. So exactly on the outside we have the sclera, then choroid, then in this ganglionic retinal layer, what we have outside the photoreceptor, middle bipolar, then inner ganglionic layer. Now what I have shown, that here in this region, what we are going to have one very important part where the light will fall. What is that spot called as? That will be called as a yellow spot. So if you can see here, this is the yellow spot, and in this yellow spot centrally it is going to have a depression. That depression is called as the fovea. So this is the fovea, and that spot is called as the yellow spot, right? Now just see here what we are going to have in the photoreceptor cell layer. So here, suppose this is the photoreceptor cell layer. So I'm going to fill with red color. This is the photoreceptor cell layer, right? So in the photoreceptor cell layer, we are going to have two types of cells. Okay, so the photoreceptor cells, they are of two types. One is called as the rods, another is called as the cones. Okay, so what are the rods and the cones? The rods are rod-like, cones are cone shaped. So depending on shape, it's given the name. The rods actually, they are sensitive to timid light. Okay, and the cones, they are sensitive to, they are working only in the bright light. Okay, they are working on the bright light only. Second thing, if it comes to the rods, that the rods the rods, they are sensitive, they are going to give us the scotopic vision. Scotopic vision means black and white. Okay, and if it comes to the cones, they are going to give us the photopic vision. Photopic vision means it is going to give us colored vision. Okay, so this is the second very important point. Then the third point, you need to know that in the rods we are going to have two components. Okay, the cell is going to have the two pigments. We'll see what are they. First of all, it's going to have one aldehyde. So what is that aldehyde? That is retinal. Okay, and here it is going to have one protein also, which is a processed vitamin A. So that's why it is said that vitamin A is good for your eye. Why? It's the reason that here these rods are going to have the pigments in it, which is made up of retinal, one aldehyde, and one protein, that is processed vitamin A, that is the opsin. Okay, so here the rods are going to have this retinal and opsin. Okay, and this opsin, this is called as rhodopsin, or rhodopsin here. Okay, so what is this? This is going to give us the scotopic vision. If it comes to the cones, it is going to have this retinal and this opsin. Okay, this opsin is called as iodopsin. So here, this one, this is called as iodopsin. Okay, Now here what is happening, what we can say, this rhodopsin is giving us a color. Okay, it's called as the visual, it's the visual purple, and it's purplish in color, and here this one is the visual violet. Okay, so remember here this aldehyde and the processed vitamin A, that is also called as the scotopsin. We can have this scotopsin. Okay, and here this opsin and retinal forms the photopsin. Okay, so what is happening here, there are some conformational changes occurs. Okay, that's how we can see different changes, and whenever we are closing our eyes, then what happens, and whenever we see light, what happens. Okay, so here this is just the structure. Now if it comes to the cones, why we can see different colors. Okay, so there are three types of cones only. What are they? They're red, blue and green. Okay, so there are three types of cones we have, red, blue and green, and we can see different colors due to the changing in the matching of, suppose, some part of red, some part of green. That's how we can able to see different colors. Okay, so after that, just see how the vision can be possible. Okay, so just see, I'm just rubbing this part. Now after that, let's just move how we can see. Okay, so first of all, how the light will fall, right? So what is happening, first of all, how the light will fall. So here what happens, we have the lens here. Okay, so first of all, the light will pass through this aqueous chamber. Obviously refraction will occur, and after that what will happen, that via this vitreous humor, here also refraction will occur, and the focusing of that light to converge on the fovea centralis, this focal length of this lens will change so that it can focus the light to the fovea. Okay, so in that fovea we have this, you can see how we have the fovea is going to have cones. Okay, so you can see, suppose here these are the photoreceptor cell layer. So this is the photoreceptor cell layer, these are the bipolar cell layer, and here the ganglionic cell layer. Okay, so first of all, suppose here these are the rods and cones we have. So as it is the fovea, it is going to have cones in it. Okay, so what are they? There are some pigments present, what I said, the opsin and retinal. Okay, so this is, suppose, the, here it is pigments, but the pigments I will show here. This is the opsin, and above the opsin we have the processed vitamin A, that is the retinal. Okay, so what is it? See, just behind the photoreceptor cell layer we have the bipolar cell layer. So bipolar cell layer, as you can remember, this is one type of neuron where we have one axon and one dendrite, and the last one is the ganglionic cell layer. Okay, so this is the ganglionic cell layer. Okay, so when light falls, then how it will fall? First of all, light will pass through this ganglionic layer, isn't it? First of all, through the inner light or inner region, what we have, ganglionic cell layer, bipolar, and then the photoreceptor cell layer. So when light falls, it will pass through ganglionic, bipolar, and then fall on the photoreceptor cell layer. The photoreceptor cell layers are sensitive to light. So here what happens, this that opsin and retinal change their conformation, and whenever there are the changes in the conformation, suppose this is the opsin and it leaves the retinal like this, so there are some changes occurs. So that's how what will happen in the photoreceptor cell layer, there are some changes occur and it will transmit. Okay, it will transmit the transmit the impulse. Okay, transmit the impulse to bipolar layer, and from the bipolar layer it will transmit this impulse to the ganglionic cell layer, and from the ganglionic cell layer it will go via the optic nerves to the brain. As you can remember, in the brain we have the center for vision. So what are the centers are there? I have told you, in the brain, in the forebrain, in the cerebrum, we have the occipital lobe. So here uh, we can able to sense or can visualize every type of images in the occipital lobe, where we can be we can process the, or we can decode the image, we can remember the images. Okay, so this is how actually the mechanism of vision completes. After that, let's just move to the next part, that there are different diseases or disorders related to the eyes. After that, let's just move to the different disorders related to the eye. Okay, so very common diseases you will see also in physics, in optometry you will get it, in optics you will get. So let's just see different disorders of the eye. Okay, so first disease we'll see here, that is myopia. Okay, so this myopia, what is that disease also called? This is called as the nearsightedness. Okay, so why it is called as the nearsightedness? See, here there are some very important thing you need to know. Everything we know about this already, that some of the people, they are myopic. So where the individual who are myopic, here what happened to the normal, what happened normally, that what happens to the near objects. Okay, to the near objects they can see normally, but the distant, okay, if it is the distant objects, it's get blurred. Okay, it's get blurred whenever it comes to the distant images. So that's why the individual can see only the near images, and if the far or the distant images they need to see, then they are getting blurred. So that condition is called as myopia. So what happens in this condition, actually, see that this is suppose the eyeball. Okay, this is the lens, this is the eyeball. Okay, so what is happening to this individual, that due to, there are two conditions that can be possible, that this is how the light should fall, okay, on the fovea. This is the normal thing. Now this individual, whenever he is suffering from myopia, this may happen that this focal length of this lens can become more and more convex. Okay, then what happens, the light will fall on the eye, but it will focus in front of the fovea. It will focus in front of the fovea. So that's why here the image formation will take place. So if it focus on this region, then what will happen, this individual can see the near images properly, but the distant images will get blurred. Okay, so what will happen to this individual, that this individual can correct his condition, myopic condition. What type of lenses need to be prescribed, that will be the concave lens. Okay, so concave lens should be prescribed, then only that individual he can correct this condition. Okay, so concave lens, okay, concave lens will be the lens which should be prescribed to the myopic individual. Now after that, the next one, the second thing, second disease, we will see what is this. So in this myopia, one question that came, that what happens to his near images. Okay, myopic individual can see the near images, that is normal, but whenever it comes to the far images, that get blurred. Okay, everything will be not blurred, only the near images he can see, but the far images he will not see. Now what will happen to this next condition, that is hypermetropia. hypermetropia. So the second is the hypermetropia. So in hypermetropia, this disease, this condition is also called as the far-sightedness. So what is happening to this individual which is suffering from hypermetropia, which is normal, near objects or the distant objects? The distant objects will be normal, but what will be blurred? The near objects will be blurred. So what is happening to that individual who is suffering from hypermetropia? That this is suppose the eyeball. Okay, so what will happen? Maybe due to the enlargement of the eyeball also this can happen, or more concaveness of the lens also it may be possible that the light will fall and focus behind the yellow spot. Suppose here the yellow spot is there, but it will be focused behind the eyeball. Okay, so behind the fovea also you can see. So this disease can be corrected by what type of lens? What type of lens should be prescribed? Convex lens. So in this condition, the convex lens can be prescribed, and that individual can able to see also the near images properly. So what is this? This disease is called as hypermetropia. After that, let's just move to the next disease. Third disease, here we will see this is the astigmatism. Okay, so what is astigmatism? So here what happens in this kind of condition? Suppose here this is the eyeball. Now due to the irregular curvature of this lens, what will happen? Or it might be the cornea also. The light will focus, but will focus in different part. Okay, so that's why this disease, what we can say it is that individual, both the near image as well as the distant images will be blurred. So in this condition, this individual which is suffering from astigmatism, this disease can be cured by some another lenses, that will be the cylindrical lens. So the cure will be the cylindrical lenses. Okay, so this is another condition here, the irregular curvature of the lens, or might be the cornea also. What happens? It will focus, but it may be in different regions. Okay, in two regions it may be focused. So that's why this individual will suffer from that condition, that both the distant and the near images they have been blurred. So this condition can be cured by cylindrical lens. Okay, so after that, let's just move to the another disease also. What is that? That is cataract. Okay, one disease is cataract and second disease is glaucoma. Okay, glaucoma, already I have told you that if the canal of Sclera gets blocked, then what will happen? The aqueous humor will start to get accumulate more and more, and that gives more pressure in the aqueous chamber. That can lead to a condition that is called as glaucoma. Second one is cataract. What happens to the cataract? Cataract, we can also call this as the Safed motif. So why this disease appears, why this disease happens? So what happens? Our cornea is transparent, right? So cornea, the cells are transparent. As we are growing older, then what happens? This cornea, the cells, we know that cells have a definite life period. Our cornea also, some cells dies and new cells are regenerating, but the new regenerated cells are also transparent. But due to the older ages, suppose some cells are died and new cells are regenerating the cornea, in that place what can happen? The new cells they are regenerating, they are not transparent, they start to get appeared opaque. Okay, and that can lead to opaqueness in the cornea. So easily this disease condition can be corrected by removing the cornea and new cornea can be replaced, right? So this is how the disease cataract can be treated. So this is how actually the diseases, different type of disorders are there. We can find a number of other diseases. You just go through all this. This is about the eye. I hope you understand. Thank you.